And we're back, everyone. So here we are for our last video on the respiratory system. Big sigh of relief, right? Excuse me. This has been quite an involved chapter. Excuse me. I realized that in recording these videos, I probably have gone a little bit longer than I would if we were in the classroom. But I think that's only because I've just been repeating things more. So I don't want you to feel too overwhelmed. You may already feel overwhelmed already. But I don't want you to feel like, oh my God, she's got 3 videos, hour long each. There's so much information. Well, it is a lot of information. This chapter does have more information than what we've covered before. But I just want to, on the backside of that, just also mention I've made these a little bit longer just because I have the benefit of recording this from my home office. I don't have the constraints of the classroom, where we only have an hour and 20 minutes. So, the videos are a little bit longer than the material usually would suggest, but it's only because I'm trying to repeat things for your benefit. Okay, enough of me jabbering on. So the first video, we talked about the anatomy. Last video, we talked a lot about pulmonary ventilation, which is breathing. We have a couple odds and ends to finish up on about or related to ventilation or breathing. And then we're going to talk about the gas exchange. We're going to talk about the oxyhemoglobin dissociation curve. We're going to talk about regulation of the respiratory system. That's going to be this final video. Okay, so we just were talking about ventilation, which is breathing. We talked about when we inhale, we contract respiratory muscles to increase lung volume, which decreases lung pressure. So when the pressure inside of our lungs drops below the pressure in the atmosphere, we develop a pressure gradient, and air moves from the atmosphere where there's higher pressure to the inside of our lungs where there's lower pressure. And then the opposite happens with exhalation. So the first odd and end I want to talk about what are some things that affect ventilation? Well, we could have some resistance to airflow moving. We could have too much alveolar surface tension. And then we also have to discuss briefly the rate at which we're breathing. Okay? All right. This is going to sound familiar because it's the same principles that we talked about in blood pressure regulation, right? And I'm sorry these squares show up, but remember, flow, whether it's fluid, blood, or whether it's air, flow is proportionate to change in pressure over resistance. So if we look at the bottom part of this equation, which is resistance, anytime we want to increase airflow, we want to decrease resistance. And then anytime we want to increase airflow, we want to have a higher pressure gradient. Excuse me. I want to talk about this later. Really, I just want to remind you, you already know this. In this case, we're talking about airflow. Airflow will be the highest. And really, we want this under most conditions. Airflow is going to be the best or highest when we have a larger pressure gradient. So now we're talking about the pressure gradient in millimeters of mercury between the atmosphere and the lungs. And we have the lowest amount of resistance. Things that can add resistance would be inflammation of the airways, like asthma, COPD, emphysema, those sorts of things. Surface tension. We've already talked about this, so I'm just reminding you. Those type 2 alveolar cells, the great cells, remember the type 2 cells that line the alveolar membrane secrete surfactant. And one of the functions of that surfactant, not only does it add moisture that we need, but it also gives us a little bit of surface tension. So just a reminder that we want a little bit surface tension to keep the lungs inflated, but not too much. So if there was a circumstance where we increase the surface tension, we can think about pneumonia leading to inflammation in the alveoli or a buildup of mucus in or around the alveoli. That would be something that increases surface tension, right? And that would be bad because if we have too much surface tension, it's going to be a form of resistance, which will reduce our airflow. But this is reviewing what we already talked about. We want to have a little bit of surface tension in the alveoli, but not too much. If we have too much, it could add resistance, and that would make it harder to move air. All right. I think I might skip this, actually, because, so don't worry about this slide. All I'd like to do here is I would like you to know these definitions. And actually, it would be pronounced V sub E. Make that in black so you can see it. V sub E or V sub A. We could talk about, and just know these definitions, we could talk about respiratory minute volume, V sub E. The amount of air moved in and out each minute. The amount that we ventilate each minute. I think that's pretty simple. The amount of air that we ventilate, the amount of air that we breathe in and out per minute. Here's a number for an average adult, but you don't need to know that number. Okay, so we can quantify what is the volume of air that we breathe in and out per minute. That's respiratory minute volume, or V sub E. But there's also a definition of something called alveolar ventilation rate. And what this refers to is how much of that air actually gets to the alveoli and will be exchanged. So the amount that reaches the alveoli for exchange. So a respiratory minute volume, V sub E, is a volume of air we breathe in and out per minute. And then we have the alveolar ventilation rate, the amount of that air that reaches the alveoli for exchange, V sub A. You can imagine a multiple choice question where you just match the correct definition. Alveolar ventilation rate, the amount of air that reaches the alveoli for exchange. Here is an average number for an adult. You do not need to know that number. However, we can compare. And look what happens. Of all the air that we breathe in, not all of it makes its way down to the alveoli. We're going to discuss that next because some of the air is lost along the way. It's going to be called a dead volume, which we'll talk about in a minute. But for now, we're just saying that we breathe in more air, but not all of it makes its way down to the alveoli for exchange. Some of the air is lost along the way. I'm going to have you know these two definitions. You don't need to know about the physiological dead space. This is the reason why not all of the air makes its way down to the alveoli. It's about 150 mils per minute, which is about the difference between that, give or take. But it just is saying that not all the air makes its way down. Some of it gets trapped in the airways. You know, we talked about the cilia that line the inner lining of the trachea. Well, not only do foreign particulates or dirt get caught in those cilia, but air can get caught in the cilia too. So this is perfectly normal. Now, if someone is losing more air than this, that will be abnormal. But we do understand that there's an anatomical dead space, which just means that there's some air that gets trapped on its way down, so that not all the air that we breathe in makes its way down to the alveoli. You do not need to know this number, but it's there just in case you're interested. Actually, I'm going to save tidal volume for lab. So in the lab, we're going to go over all of the different volumes and capacities. Tidal volume is one of them. So we'll save that for lab. All right, enough of that. Let's talk about gas exchange. Way back at the beginning of this chapter, we kind of broke down the functions of the respiratory system in order. And we said that, well, first, we've got to bring in the air. And we talked about that. That's ventilation. We bring in the air. It goes all the way through the airways down to the alveoli. And then the number 2 thing that happens is we have external respiration, which is gas exchange between the alveoli and the surrounding blood vessels. So, you should have a definition. You should know that external respiration, sometimes called alveolar respiration. I would make sure you know both definitions, or both terms, because I could use either. I think it makes sense that it's called alveolar respiration because it occurs at the alveoli. So it's gas exchange between the alveoli and the surrounding blood vessels. And then also make sure you know which direction each gas is moving. And I tried my best to use arrows and colors in this bottom picture. And it's also in words. Here, oxygen moves from inside the alveoli, across the alveolar membrane into the capillaries. And then CO2 moves from the bloodstream back into the alveoli. So imagine these as arrows. I think sometimes people get confused with these, but it's just memorization. So I think if you spend enough time with it, you can get it down. Now, we're going to talk a little bit more about this gas exchange. One thing that's important is that these gases must first dissolve in fluid before they diffuse across the alveolar membrane. And I already talked about this, remember? When I introduced the great type 2 cells, and I said that they secrete surfactant. One of the jobs of the surfactant is to keep the lung inflated. One of the jobs of the surfactant is to provide some surface tension, but not too much. And then the other job of the surfactant is to provide fluid, because we need fluid here. The gases will dissolve in water before they diffuse across the membrane. There is a law about this, but I think I've given you enough laws to know with Boyle's law and Dalton's law, so let's not worry about Henry's law for now. I would like you to know this concept at the very bottom, but you don't have to know the specific name of Henry's law. Okay, so what are we saying? We are saying that we have external respiration, gas exchange between the alveoli and the surrounding bloodstream. Oxygen will diffuse across from the alveoli into the blood. Carbon dioxide will diffuse from the blood back into the alveoli, so it can go up the respiratory tract and we'll breathe it out. Before either of these gases diffuses across the alveolar membrane, it dissolves in the fluid provided by the surfactant. This process, the amount of gas that is able to dissolve in that water, is really determined by 2 things. And I put them in yellow because we're going to talk about them. Well, how much gas dissolves is influenced by that particular gas's solubility in water, but also how much a gas dissolves in that water, how easily it dissolves in that water, is related to the partial pressure that is there. And we're going to talk about this. Okay, here is just more information. This is the same picture I showed you before. We have external respiration, gas exchange between the alveoli and the surrounding bloodstream. I said that oxygen will move from the alveoli into the bloodstream. Carbon dioxide will move from the bloodstream back into the alveoli. Excuse me, I have to take a sip of water here. So This diffusion, so first of all, diffusion is the mechanism at work. There's really nothing fancy. Back in A&P1, we had a whole lab on osmosis and diffusion, and you probably hated it. But we did that because diffusion and osmosis are the forces behind so much of what happens in our body. If we can create a concentration gradient and we can put between two sides of that concentration gradient a semipermeable membrane, then we know that diffusion will happen. And that's what happens here. We have a concentration gradient where there's more of one gas on one side versus the other. Think about oxygen. There's going to be more oxygen in the alveoli because you just breathed it in. So there's more oxygen in the alveoli compared to the blood. So oxygen will move from a higher concentration inside the alveoli to a lower concentration in the blood. CO2, we're going to have more CO2 in this venous blood here. Very little CO2 inside the alveoli. So CO2 will move along this concentration gradient from higher amounts in the venous blood to lower amounts inside the alveoli. And I'm going to give you some of the pressure differences. So let me fill in the gaps here. So if we're looking at oxygen, the partial pressure of oxygen in the alveoli is 100 millimeters of mercury. The partial pressure of oxygen in the surrounding capillaries is only 40. So there's some oxygen here, but not much. You do not need to know these numbers. You don't need to know these numbers. I'm just giving you them as examples to demonstrate that there is a higher concentration gradient here and a lower concentration gradient here. So that's why oxygen will move from high to low. On the other hand, there is a very lower partial pressure of carbon dioxide in the alveoli versus the partial pressure of carbon dioxide in the capillaries. What I want to show to you in looking at these numbers, even though you don't have to know the numbers specifically, I'd like you to look at the trend. Well, obviously, we have a concentration gradient, but there's a bigger concentration gradient for one gas compared to the other. So have a look. Is there a bigger concentration gradient in oxygen or carbon dioxide? There's a bigger concentration gradient for oxygen, right? Oxygen's going from 100 to 40, whereas carbon dioxide is going only from 45 to 40. So both have a concentration gradient, but the gradient is higher for oxygen. And we're going to talk about this in a minute. Okay, you're going to see this language in the textbook and other places. At the alveoli, blood unloads CO2 and loads O2. I don't love that language because it doesn't tell me where the gas is going from where to where. So I will not be using that language on any test questions, but I just wanted to point it. I mean, it does make sense, but I think for maybe first learning it, I think it's easier to just talk about, oh yeah, oxygen is moving from alveoli to blood. Carbon dioxide is moving from the blood to the alveoli. We're just filling in the gaps. At the very beginning, we said what's the first thing we have to do is ventilate. And then we bring that oxygen to the lungs where we do external respiration. Okay. What are some of the factors that influence this gas exchange? We talked about factors that influence ventilation. What are some factors that influence how oxygen and carbon dioxide move across the alveolar membrane? Well, one of them is concentration gradient. And as I said, the concentration gradient is higher for oxygen. So it begs the question, well, don't we want to be exchanging about equal amounts of these gases? And the answer is yes. So what we're finding out here is even though we need both gases to have a concentration gradient and we need both gases to be soluble in water, oxygen and carbon dioxide benefit more from one than the other. So even though both have a concentration gradient and are soluble in water, we see that oxygen benefits from a higher concentration gradient. It's not as soluble compared to carbon dioxide. But carbon dioxide is much more soluble in water than oxygen. So this means that the exchange of gases more or less evens out. We're exchanging a similar amount of oxygen versus carbon dioxide in opposite directions. It's just that oxygen is more reliant, benefits more from a higher concentration gradient, and then CO2 is able to keep up because it, even though it has a lower concentration gradient, it is more soluble in water. This is pretty simple membrane thickness. We ideally like to have a thin alveolar membrane. If that membrane gets thicker, it would impede gas exchange. The more membrane surface area we have, the more gas exchange we can do. This is pretty common sense stuff. I'm not going to ask you that on a test. There is something called the ventilation perfusion ratio. And this means that we need to be able to match our ventilation with our perfusion. Ventilation we've already talked about. Ventilation is the amount of air that we breathe in. And actually, if I can remind you, even though you don't have to know the numbers, we said that the minute ventilation was about 6 liters, but only about 4.2 of those liters made it to the alveoli. So that's why we're seeing down here, our ventilation is about 4.2 liters. So we have about 4.2 liters of air reaching the alveolar membrane, but we perfuse it, which really means the amount of gas that is being exchanged. We perfuse it at a higher rate. So that's all I'm saying here. Ventilation is the amount of air that gets to the alveoli. It's alveolar ventilation, really. So for whatever volume of air that gets to the alveoli, we perfuse or exchange it faster. And this would be the normal ratio. So indeed, if you divide 4.2 times 5.5, you'll get 0.8. And that's our usual VQ ratio, or ventilation perfusion coupling. And this is normal. I'm not going to ask you those numbers, just that one is higher than the other. For whatever flow of air reaches the alveoli, the rate of perfusion is higher. Okay, so what's our third step? Well, we ventilated, we brought in the air all the way down to the alveoli. Step number 2 is we do external respiration, where we transfer through diffusion the oxygen from the alveoli into the blood. And of course, at the same time, diffuse carbon dioxide from the blood back into the alveoli so we can breathe it out. But now we have this oxygen-rich blood. It's going to be pumped back to the heart, and the heart is going to pump out that oxygen-rich blood out to the body. We already know, how do we transport oxygen? On hemoglobin. So we already know that. Of course, on the venous side, transporting carbon dioxide, we can transport some carbon dioxide that's dissolved in plasma or bound to hemoglobin. But what I want to introduce to you, because we're going to see it later, the primary way that we transport carbon dioxide, particularly in the venous blood, is as part of the bicarbonate ion. And you can see I put in yellow the most important things to help, since we have so much going on here. So most of our CO2 gets transported, kind of hitches a ride in this bicarbonate ion. Straightforward. What happens once that oxygen-rich blood gets to our tissues? We have our last gas exchange called internal or systemic gas exchange. So also know it's called systemic or internal. This is gas exchange between the blood and the tissues. Same principles. It's diffusion. I'm going to give you some numbers, but you don't need to know the numbers. In case you're wondering, oxygen is going to move from the blood into the tissues. Could be a muscle, could be an organ. Carbon dioxide is going to move from the tissues back into the blood. Let me say that again. Oxygen moves from the blood into the tissues. Carbon dioxide moves from the tissues back into the blood. Carbon dioxide is going to benefit from a greater concentration gradient. The partial pressure of oxygen in the blood is 95 millimeters of mercury. Versus in the surrounding tissues, it's 40 millimeters of mercury. On the other hand, the partial pressure of CO2 in the tissues is 45, and then the partial pressure of carbon dioxide in the blood is 40. Both have a concentration gradient, but the concentration gradient is greater for oxygen. How does CO2 keep up? Because it's more soluble in water. And this is involving the blood, so we can benefit from these gases dissolving in blood. Okay. All right, my friends. Now, this is the last hard part of this chapter. But the good news is I'm here to break it down to you. And I've put in yellow some really important things. I have even put on Blackboard a handy-dandy worksheet for you. I believe it might be even called the best ever explanation of the oxyhemoglobin dissociation curve. This curve tells us a lot of information, and even though you might be kicking and screaming, I'm going to walk you through it because I know you can do it. So I'm going to walk you through this in steps that I believe would be the easiest way to understand this. So let's first talk about, well, what is this curve? Well, on the x-axis, we have the partial pressure of oxygen. Another way of saying that on the x-axis is really the amount of oxygen. The amount of oxygen dictates its partial pressure. So it might be easier for you when you're first trying to grasp this to just think of the x-axis as the amount of oxygen. So the x-axis is the amount of oxygen, and then the y-axis is hemoglobin saturation. And of course, we mean hemoglobin saturation with oxygen. So we're comparing the amount of oxygen that's present with how much of that oxygen is able to bind to hemoglobin. This is a direct relationship. The more oxygen we have available, then we can more fully bind hemoglobin with oxygen. I mean, I think that's really simple. We know that the more oxygen that there is in the blood, the better able we are going to be to fully saturate hemoglobin. Remember, each hemoglobin can hold 4 oxygens? Well, if we have a lot of oxygen, we can fill more hemoglobin. And the curve shows us that. At the beginning, when we don't have that much oxygen, we don't have that much hemoglobin saturation. But once we are able to provide more oxygen, well, then we can fully saturate hemoglobin. Okay. So understand just what it is. The next thing to understand is, why is there such a sharp curve or a steep slope at the beginning of this graph? Because ultimately, it could just be a line that looks like that, but it doesn't, right? So why do we have that really steep slope at the beginning? And the answer is right here. What happens is, we know that hemoglobin can hold a maximum of 4 oxygens. So if we have hemoglobin, it can bind 1, 2, 3, 4, right? Well, what happens is, once the first oxygen binds, the remaining oxygens can bind much more quickly. So at first, it's a little bit of a slow reaction. It takes some time to get the first spots on each hemoglobin bound with oxygen. But there's a molecular thing that happens. The shape of hemoglobin changes. It's a molecular thing that after the first hemoglobin binds, the remaining 3, boom, 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 can bind so quickly. So that's why we see that the rest fill up so quickly. And we very quickly get to full saturation. So, why is there a sharp curve in the graph? Because at the beginning, it takes a little bit of time to fill the first spot on hemoglobin with oxygen. But after we fill that first spot, the remaining 3 oxygens are able to more quickly bind to hemoglobin. Pretty cool, right? Good. Let me add one here. Why does the curve level off? Why doesn't it keep going up here? Hopefully you're already thinking with me on this. Well, the reason it levels off is because once a hemoglobin is fully saturated, there's no more spots available. So 100% saturation means that all spots are taken. So the curve will level off at 100, because once all 4 of those spots are taken, even if there's more oxygen present in the blood, there wouldn't be any spots available for them to bind to on hemoglobin. Good. All right, the last point on our discussion of the oxyhemoglobin dissociation curve. This curve also shows us something really cool. It shows us that we can have a pretty significant drop in oxygen in the blood and still maintain a pretty high hemoglobin saturation. Let me say that again. Our body can handle, this right here is showing us a 40% from a partial pressure of 100 of oxygen to 60. We can, that is pretty low, but I'm just making a point. We can handle a drop in oxygen levels and still keep hemoglobin well saturated. Or you can also say it this way. Normal oxygen transport on hemoglobin can still occur even at smaller amounts of oxygen. And this is good. This gives us a little bit of a buffer. If our cells need oxygen and we don't have enough right away in the blood, it gives us a little bit of buffer that our hemoglobin is still going to have some oxygen to give. So let's just give you 2 examples. Look at the yellow. And I gave you this number before. You don't have to know it, but normal alveolar partial pressure of oxygen is 100, right? So at a partial pressure of oxygen of 100, which means when we have a lot of oxygen, we will have 97% hemoglobin saturation. Isn't it almost at 100? In fact, When we do the hemoglobin saturation, which you should know this, right? The pulse oximeter, you put it on someone's fingertip, it sends these light rays through the skin, because if hemoglobin is saturated more, if you shine a light through it, it'll be more dense. Well, the normal is really close to 100. But you'll still be living if it goes down to 90. We don't like that, but you'll still be living. So at our normal alveolar PO2, we've got 97 at least percent hemoglobin saturation. But look, even if I drop the amount of oxygen to 60, that's a 40% drop. Even if I drop the amount of oxygen 40%, I'm still going to be at 90% hemoglobin saturation. So this gives us a bit of a buffer. Even if we, in a period of time, have a drop of oxygen levels in the blood, up to a certain point, we can still maintain hemoglobin saturation fairly well. What I want to point out to you is this is not true of carbon dioxide. We cannot have an increase in carbon dioxide by 40% and be living. No. I think I said this in one of the videos earlier. A good gauge is if we have a 10% increase in CO2, our respiratory rate will double to try and get rid of that, because excessive CO2 makes the blood very acidic very quickly. So, the idea that we have a little bit more play with oxygen because of what this curve shows us, because of the affinity for oxygen to hemoglobin, because it can bind so quickly, the second, third, and fourth one, it gives us a little bit of room for error. And we don't have that with carbon dioxide. So as we're going to find out later this semester, our body has to do a lot more things to try and handle excess CO2. We don't have to do as many things to try and handle a lack of oxygen, because this curve shows us that we can handle a little bit of a drop in oxygen for a period of time. And then we'll increase respiratory rate to bring in more. And then by the time you need it, it's down. I'm not saying that's always the case, because of course we can have conditions where you don't have enough oxygen. But this is true of oxygen, not true of carbon dioxide. Okay, now there's two situations I want to talk about of how this curve changes. Now, I know I'm asking a lot of you, I know that, but I'm asking a lot of you because I know you can do it. So let's just review. Well, first of all, what does this curve show us? Well, it relates the amount of oxygen present to the amount of hemoglobin saturation with oxygen. We know that there's a direct relationship. As we have more oxygen available, we have more hemoglobin saturation. Then we talked about, why is there a sharp curve at the beginning? Well, that is because after the first oxygen binds to a hemoglobin molecule, there's a molecular change which allows the remaining second, third, and fourth oxygens to bind to hemoglobin more quickly. So hemoglobin will fill up very quickly. We talked about why does the curve level off? Well, because once all four spots on hemoglobin are taken, even if we have more oxygen present, there's no more spots on hemoglobin. Then we talked about this curve shows us that even if we have a pretty significant drop in oxygen, we can maintain pretty high hemoglobin saturation. Of course, not past a certain point. The magic number seems to be about 60 millimeters, or 60, a PO2 of 60. But we can handle a pretty significant drop in the amount of oxygen present and still maintain decent hemoglobin saturation. Okay, so I want to talk about two examples of how this curve will change. One of them is temperature, and one of them is pH. Bear with me. Let's give an instance where we have an increase in core temperature. So look at the blue line. The blue line is our normal oxyhemoglobin dissociation curve. Blue is normal. When we are in an environment where the temperature in our body goes up, the curve shifts to the right. When the temperature goes up, right, at normal body temperature, 38 degrees Celsius, normal blue curve. When our temperature rises, now it's 43 degrees, look at our red line. Now the curve shifts to the right. When the curve shifts to the right, we have more oxygen unloading. Or what that means is when the curve shifts to the right, hemoglobin releases more oxygen. So, like we said, if we're at a PO2 of 60, the normal hemoglobin saturation would be about 90. But what about at that same PO2 of 60 for the blue line? Now I'm at a hemoglobin saturation of maybe 75. So when the curve shifts to the right, hemoglobin is releasing more oxygen, which means that hemoglobin saturation goes down. But it's not always a bad thing. I give you an example of exercise. When we have an increase in body temperature, when we're exercising, it's actually helpful for our hemoglobin to release more oxygen because our working muscles are going to need it. Now, if we have a fever and our body has an immune response, systemic immune response going on, that may not be a good thing. Now, it may be a good thing because maybe our immune cells need more oxygen to do their job. So just because hemoglobin saturation goes down doesn't always mean it's a bad thing. In fact, it's going down for a reason. In this case, it's going down because there's something happening in the body that is increasing our core temperature, and your body is responding by delivering more oxygen to the tissues to try and help whatever it is that is causing our elevated temperature. Now, of course, at a certain point, this could be bad, right? We don't want hemoglobin to release too much. Okay, great. So this last part, we're talking about how this curve changes under two conditions. Our first condition is an elevated temperature. When we have an elevated body temperature, the curve shifts to the right. When the curve shifts to the right, hemoglobin is releasing more oxygen to the tissues. Therefore, hemoglobin saturation goes down for the same given PO2. Good. Now, what we're going to find is the same shift, a shift to the right, when we have a drop in pH. And I picked these two instances because they're the more common, right? It's more common for us to have an increase in core temperature for a certain reason. It's more common for our blood to be faced with an acidic situation. So, also, when our pH of the blood goes down, guess what? The curve shifts to the right. When the curve shifts to the right, more oxygen is released, which means that hemoglobin saturation goes down. But here's the reason why, and I'd like you to know this. The reason why the hemoglobin releases, let me say this again. The reason why the curve shifts to the right when our blood pH drops is because when the curve shifts to the right, more oxygen is released. We may recall that when our blood is acidic, often that is caused by too much CO2 in the blood. So if that's the problem, couldn't that be helped by releasing more oxygen in the blood? So by releasing more oxygen, it's counteracting the effect of too much CO2. What's happening is, at that given point in time, the body can't immediately get rid of CO2. There are ways that we're going to be able to do it, but in that moment, immediately, the body can't get rid of that elevated CO2. So in the meantime, before we can do other things to get rid of the CO2 permanently, we can purposely release more oxygen from hemoglobin. And that will counteract the effect of too much CO2 temporarily. So in both cases, when we have an elevated core temperature and we have a drop in blood pH, the curve shifts to the right to release more oxygen. There you go, folks. You have gotten through maybe the hardest part of this chapter. Believe it or not, the end here is not going to be as difficult, so let's just get through it. As you're going to see, for every body system that we go through, we have to talk about the regulation of that body system. And no different when we talk about the respiratory system. One thing that's different between the respiratory and cardiovascular systems, the respiratory system has no internal control mechanism. I mean, so these are things that are unique to the respiratory system. You know, the cardiovascular has its own little internal regulatory system. We had the SA node that can help regulate nerve impulses right there. Well, the respiratory system doesn't have a same internal regulatory center. And also what's unique about the respiratory system is that we actually have some voluntary control over breathing. You do not have any voluntary control over heart rate. I mean, sure, people can train themselves to maybe lower their heart rate a little bit. But, I mean, with the respiratory system, any one of us can just say, hold your breath. You can't just say, oh, automatically stop your heart. So that's something that's unique. So we're going to talk about 3 ways in which the respiratory system is regulated. We are going to see the control centers in the brain. So the brainstem, the medulla, and pons. And this is not, I mean, if we think back to the cardiovascular system, the medulla was a main cardiovascular regulating center. The medulla is also a major regulating center of the respiratory system, but the respiratory system also has help from the pons. In order to be able to regulate, the medulla and pons gets input from peripheral receptors, which is really happens all the time in every body system. And we do have an ability to do some autoregulation. Okay, I've already talked about this. We do have some voluntary control. This is extra information. All I want to say, though, is we have some voluntary control, but ultimately, our involuntary controls will take over. For example, if I tell you to hold your breath, you're going to be able to hold your breath for a period of time. At some point, even if you voluntarily still want to hold your breath, your body will take over. So whether that means that you'll just start to breathe again on your own, or if you're really die hard and you keep holding your breath, your body's going to pass out. It's kind of like when someone drinks alcohol. They drink so much alcohol that their body just passes out because that's the only way the body knows how to get through to you, right? Because when you're passed out, you can't put more drink to your mouth. So we have some voluntary control, but ultimately, our involuntary controls will take over. Okay, this is going to be talking about some of those peripheral receptors. Let me just peek ahead for a minute here. Okay, good. All these are true. I'm not going to talk about these specifically. In the next couple of slides, I will get into some specifics. I do want to remind you, I would like you to know the two parts of the brain that are our main respiratory control centers are the medulla and pons. Now, when we talk about those peripheral receptors, by far the biggest thing that trips our medulla and pons to regulate our respiratory rate is blood pH. So 2/3 to over 2/3, 3/4, what am I talking about? 3/4 of the medulla's actions to regulate respiratory rate come as a result of pH changes. Okay, so I'd like you to know that. I did give you an example. When our CO2 in our blood goes up, so when blood CO2 goes up and blood becomes acidic, the medulla will respond by increasing our respiratory rate. That is the primary way that we get rid of carbon dioxide. Remember I said that if your blood CO2 levels go up by 10%, your respiratory rate will be doubled. And that action will be led by the medulla. There you go, right? A rise in CO2 of 10% causes our respiratory rate to double. Of course, we do have some influence from oxygen, right? So at some point when oxygen levels drop too much, our medulla will become active to fix it. But because of the oxyhemoglobin association curve, we can withstand pretty significant changes before the medulla kicks in. So our medulla, even though it's going to be responsive to changes in oxygen levels and CO2 levels in our blood, it's much more responsive to CO2. I'd like you to know some of the brain control centers. I've put in yellow what you need to know, the medulla and the pons. You don't have to know the specific names. I mean, basically what happens is we have these clusters of nuclei in the medulla and pons. And each little cluster of nuclei is responsible for overseeing a different thing. I'm trying to simplify it as best I can, which is really hard. So one example is in the medulla. We have a specific cluster of nuclei that really help to regulate our inspiration or inhalation. Of course, there's more to it, but this is one specific I can point out. The pons might be more active in our shifting from one to the other. They work together, of course, the medulla and the pons. It's much more complicated, but I've just tried to give you a little taste. So medulla and pons, one function of the medulla specifically when it comes to regulating the respiratory system is that it really controls our inhalation or inspiration. And one specific function of the pons is that it helps when we shift from one to the other. We don't really need to go over this one, even though I have it written down, so you review it. We already talked about it. Those bronchioles. Now we're going way back, right? The The bronchioles, because they're covered with smooth muscle, they have the ability to constrict or dilate. If we need more airflow, what will happen to the bronchioles? If we need more airflow, they will dilate. If we're sleeping and we don't need as much airflow, then the bronchioles will constrict a little bit. Okay, few odds and ends, and then we'll get you out of here. I would like you to be familiar with two categories of respiratory system disorders. For many of you who are working, as PCAs, or, I teach an A&P class for EMTs. They see, because I don't work as an EMT or a PCA, they tell me, and I read about it, they see a lot of respiratory distress. When I poll the EMTs, they tell me if they had to pick one thing that they happen to get called for the most, it's for respiratory distress. Really, really common, whether it's actually a specific respiratory problem or it's something else that's problematic, but it's just showing respiratory symptoms as the body's trying to handle it. Well, we can actually be a little simple about it. And almost all respiratory problems can be categorized as either restrictive or obstructive. Let's start with obstructive. And the name gives it away. An obstructive respiratory disorder is one where the airways are obstructed. Narrowing or blocking of the airways. Airways are obstructed or restricted. What are some examples of this? Asthma. Asthma is chronic inflammation of the airways. If you usually have a nice open airway, nose, pharynx, trachea, bronchi, bronchioles, if you usually have a nice open airway, well, then it's easy to exchange, to bring in and push out gases. If that airway is for some reason narrowed in asthma, it's inflammation. The inner lining of the airways are inflamed. So the actual opening of the airway is a little bit smaller in diameter. And that makes it more difficult to breathe in and breathe out. Another example of an obstructive disorder is COPD, chronic obstructive pulmonary disorder. It's in the name, chronic obstructive pulmonary disorder. And I do want to point something out. Both in asthma and COPD, often the hardest thing is breathing out. So if you have a person with asthma who's having an asthmatic attack or COPD, it's harder to breathe both directions, but in particular, it seems to be a little bit harder to breathe out. I've given you other examples, but I've circled the ones that I might use on a test. And then if a disorder is not obstructive, it's most likely restrictive. And the fancy word for this is that it reduces pulmonary compliance. But what it has to do is it limits how much the lungs or alveoli can be inflated. And as a result, it limits gas exchange. So the problem in a restrictive disorder isn't that the airway is compromised. The problem is actually in the respiratory zone, places that we're trying to exchange gases. When you see pulmonary compliance, compliance means the ability for something, in this case, the alveoli. Compliance means that the alveoli can fill with air and then recoil a bit. Fill with air and recoil a bit. They're responsive to that. They're compliant. They're able to expand a little and then come back. Expand a little and then come back. If something reduces pulmonary compliance, for whatever reason, the alveoli aren't able to expand as much, or they're not able to recoil as much. The biggest example of this is probably pulmonary fibrosis. And that word fibrosis kind of gives it away. In pulmonary fibrosis, what happens is instead of the alveolar membranes being nice and, I hate to use the word supple, but the alveolar membrane should be kind of supple. The alveolar membrane should be moist, and they should be compliant. They should be able to fill with air and then recoil. Well, in pulmonary fibrosis, the alveolar membranes become fibrotic, hard, crusty, no longer compliant. And if we can't move the alveolar membranes in and out, if we replace a nice smooth alveolar surface with hardened scars, well, you can't exchange gases. All right, cool. I mean, it's not cool to have that, but we can see on x-ray, you can see some of the scarring from pulmonary fibrosis. Here's an actual lung taken out. This is my annual Don't Smoke campaign. So an emphysema is actually considered an obstructive disorder, but it kind of wears away the alveolar membranes. Well, if it wears away, you can't exchange gases, and then they become scarred. Normal lung on the left, smoker's lung on the right. Don't do it. This is extra. You don't need to know it. Don't need to know that. Let's see. Okay. I would like you to know some of these terms, if you don't already. Hypercapnia means too much CO2. Hypocapnia means too little CO2. I mean, we can make this really simple. If somebody is hypercapnic, too much CO2. Hypocapnic, too little CO2. Hypoxia is a drop in oxygen at a tissue. So let's see, hypoxia, too little oxygen at a tissue. Anoxia would be no oxygen at a tissue. Hyperpnea is when ventilation rate goes up. Dyspnea is just labored breathing. I'm just going to ask you these top three. All right, the volumes and capacities we're going to cover in lab. So I'm going to save it for lab. I do go a little crazy on a lot of this. You don't need to know these end slides. In lab, we're going to go through this. We're going to go through the volumes and capacities that I would like you to know, but I'll save that for lab. All right. Listen, work hard. You can absolutely do it. Take the time. I'm here if you have questions, okay? All right, good luck, everyone.